Okay, so welcome to this next video in which we are discussing angiogenesis in chronic inflammation. Okay, so we've seen that the angiogenic factors released by the T cells and the macrophages will activate these receptor tyrosine kinases, these vascular endothelial growth factor receptor 2s and these fibroblast growth factor receptor 1s, and they will now have these phosphotyrosine residues. Okay, now the phosphotyrosine residues had led to the assembly of these Ras-Jef complexes on their surface, and that had led to a whole pathway which led to the activation of two transcription factors, the MYC transcription factors and the C-phos-C-jun heterodimers. Okay, but now what we want to look at is a totally separate pathway which is going to start with the phosphotyrosine residues and is going to lead to the activation of a totally different transcription factor, which is going to actually do the same thing as MYC and C-phos and C-jun, and is therefore also going to contribute to this increased proliferation, which we'll discuss therefore all together once we've got all three pathways side by side. Okay, so we'll start off with our receptor tyrosine kinase dimer here. So we'll draw the one for the fibroblast growth factor receptor 1, simply because it has fewer of these immunoglobulin-like domains and is therefore slightly simpler to draw. Okay, so here is the two portions of the tyrosine kinase domain. Okay, here is the phospholipid bilayer, and of course the receptor has dimerized, so we'll draw the fibroblast growth factor 2 dimer here. Okay, and then here is the other fibroblast growth factor receptor 1 here. Okay, here is the transmembrane region, and here is the split tyrosine kinase domain down here with the kinase insert there. Okay, and you've now got a bunch of phosphorylated tyrosine residues in this kinase insert region and also in this tail after the second portion of the kinase domain. Okay, so I'll highlight these in purple. And now what these are going to activate is an enzyme known as phospholipase C gamma. Okay, so phospholipase C gamma is going to be activated by these phosphotyrosine residues. Okay, and it's often abbreviated to PLC gamma. So this stands for phospholipase C gamma. Okay, so this is a different type of phospholipase C than the usual type that you would see in most pathways. So in the GQ pathway, which is a very famous pathway, more famous than this anyway, which is a G protein coupled, it's a G protein pathway that's downstream of G protein coupled receptors. Okay, you activate a phospholipase C enzyme, but you don't activate phospholipase C gamma. You usually activate phospholipase C beta. In this case, these phosphotyrosine residues are going to activate an enzyme known as phospholipase C gamma. And what's going to happen is that phospholipase C gamma is going to start breaking down a usual component of the phospholipid bilayer, which is called PIP2, into diacylglyceride and IP3. Okay, so let me just discuss what PIP2 is. So, if this is the phospholipid bilayer, then the phospholipid bilayer is made up of phospholipids. So let me show a phospholipid, and then I'll show how to modify a phospholipid to get a molecule of PIP2. So what we're heading for is PIP2, which stands for phosphatidyl inositol 4,5-bisphosphate. Okay, so this is phosphatidyl inositol and then 4,5-bisphosphate. So the initial P is for phosphatidyl, the I is for inositol, and then the P2 on the end is for phosphate, and then since you've got two phosphate groups, it's P2. So that's phosphatidylinositol 4,5-bisphosphate, or PIP2, also often just referred to as PIP2. Right, so the structure that I have drawn in cartoon form down here is a standard phospholipid. So it has the two long-chain carboxylic acids, which are esterified to the first and second alcohol groups of the glycerol molecule, which is the backbone for the phospholipid. Okay, so these are also known as fatty acids. So fatty acids is the biochemist's name for a long-chain carboxylic acid. Okay, and they are esterified to the first and second alcohol groups of what I've now shaded in green. This horizontal line represents the glycerol backbone of the phospholipid. Okay, and the correct, or the chemist's name for glycerol is to call it propane 1,2,3-triol. Okay, and although this is a mouthful, it is actually quite useful because glycerol doesn't tell you at all what the molecule actually is, whereas propane 1,2,3-triol tells you exactly what this green molecule is. It's a 3-carbon molecule where every single one of the carbons has an alcohol group coming off it. Okay, and of the first and the second alcohol groups you have esterified fatty acids, and of this third alcohol group you then have a phosphate group. So in pink here, this is a phosphate group. Okay, right. Now, the entire structure is now what's known as a phospholipid, or indeed, the old name for a phospholipid was to call it a phosphatidate molecule. And although no one will ever call, well, you'll never hear anyone call a phospholipid a phosphatidate molecule, when we are talking about modified phospholipids, we use the term phosphatidate all the time. For instance, up here, phosphatidyl, that's what this means. It means one of these phospholipids. And what I hope to convince you of is that PIP2, or PIP2, is just a modified phospholipid. So let's now draw the structure of PIP2. So we start with this exact same molecule again, this phosphatidate group here. Okay, or this phosphatidyl group. So what we're going to do is we're going to need to create phosphatidyl inositol. Now, how are we going to create phosphatidyl inositol? Well, it's simple. We just link an inositol molecule onto our phosphatidate molecule. Okay, so what is an inositol molecule? Well, basically, an inositol molecule is a 6-carbon ring. So let me draw its skeletal structure. So remember, in skeletal structures, you don't show carbon atoms. They're implicitly shown as they're implicitly shown as corners. Okay, and this is to make the structure easier on the eye. Okay, so these 6 corners of this hexagon implicitly represent carbons. Now, of every single one of the carbons in the inositol ring, what you have is an alcohol group coming off. Okay, so it's an incredibly rotationally symmetric molecule. So you have 6 alcohol groups in total, and then you'll notice that each of the carbons in this structure, so for instance if we take this one here, it only has 3 bonds, 1, 2, 3, and because we're drawing a skeletal structure, if we have missing bonds of carbons, it's implicitly assumed, it's implicitly understood that the final bond is to a hydrogen atom, so you don't show bonds of carbons to hydrogen atoms. And this is why it makes the structure easier on the eye, because if you were trying to fit in hydrogens as well, it would look a right mess. So this is the structure of inositol. Okay, so what we're going to do is we're going to take this inositol molecule and we're going to link it up to the phospholipid molecule by forming a phosphoester link between one of the alcohol groups on the inositol molecule and the free alcohol group of the phosphate group of the phosphatidate molecule. So we're going to link inositol off of this phosphatidate molecule here. So I will just draw inositol as a hexagon here, which I will colour in blue. And when you do this, what you get is a molecule of phosphatidyl inositol, or PI. Okay, so that's this first bit of this name sorted. But we don't want phosphatidyl inositol, we want phosphatidyl inositol 4,5-bisphosphate. So we now need to add 2 more phosphate groups onto this structure. So we just need to label up the carbons of the inositol ring now. So we label this one that's already attached to a phosphate group number 1, and then we work round rotationally. So we label this 2, this one 3, 4, so we're going to add a phosphate group onto the alcohol group of that carbon, and then 5, we'll add another phosphate group on there, and that gives us phosphatidyl inositol 4,5-bisphosphate. Okay, right, or PIP2. So you can see that this is just a phosphatidate molecule, it's just a phospholipid with a bigger head basically. So you can insert this into the membrane just like a normal phospholipid, so I'll put it as though it's in the membrane, and all that's going to change is that it's got a bigger bit sticking into the cytoplasm, or a bigger bit sticking out into the extracellular space basically. Okay, so this is a normal component of the phospholipid bilayer. You don't just have these boring phospholipids, you also have some variation. You'll have these ones as well. Okay, so when you activate the phospholipase C gamma, what it's going to do is it's going to start breaking down these molecules that are within the phospholipid bilayer. And all phospholipase C enzymes break down PIP2, and they all cut this bond here. Okay, they cut the bond between the glycerol molecule and this phosphate group here, and they therefore create 2 products. So they create glycerol now linked to 2 fatty acids. So here's the glycerol in green, and we have these 2 fatty acids bound to the glycerol in orange here. And this molecule is what's known as a diacyl glyceride, okay? Or for short, it's called DAG. So this is DAG. And the reason for that is that it's a glycerol molecule, so that's the glyceride bit, and it's got 2 acyl groups attached to it. So let me explain what an acyl group is. So basically, the free fatty acid would be a long-chain carboxylic acid, so it would have this structure here. However, when you esterify it to an alcohol group on glycerol, what happens is you take off that alcohol group from the carboxylic acid group, you take off the hydrogen from the alcohol group on the glycerol, you combine those together to make water, okay? So off will come water, and that's why it's called a condensation reaction. Then what you do is you bind this carbon of the carboxylic acid to the oxygen, and that creates you the ester link between the carboxylic acid or the fatty acid and the alcohol group of the glycerol. However, you can see that we haven't actually added on the full carboxylic acid. Instead, we've only used this portion here, and that portion is known as the acyl group. Okay, so we actually have just added on 2 acyl groups onto the alcohol groups of our glycerol molecule in the first and second position. So that's why it's a diacyl glyceride. It's glycerol with 2 acyl groups attached to it. Okay, and the other important molecule that you get is this molecule of inositol with these 3 phosphate groups attached to it here. Okay, so here's the hexagon of inositol again, and it's got phosphate groups coming off it, off the 1st, the 4th, and the 5th carbons here. Okay, so we'll colour in the inositol ring again in blue, and then we'll colour in these phosphate groups in purple again. Okay, now this molecule is what's known as inositol, which makes perfect sense, and then 1,4,5 trisphosphate, okay? So we're just labelling up which carbons the phosphate groups come off, and then we're just labelling it trisphosphate because we've got 3 phosphate groups now coming off the inositol ring. Now, because inositol 1,4,5 trisphosphate is a bit of a mouthful, people will often just abbreviate this to I for inositol, P for phosphate, and then they'll put a 3 there to denote that there are 3 phosphate groups coming off of the inositol. Okay, now, we're not interested in what the diacylglyceride does. That remains in the phospholipid bilayer after you've done this breakdown, which again makes sense because you've cut effectively the head of the phospholipid off, and the tail bit is going to remain in the phospholipid bilayer whilst the head goes off into the cytoplasm. We're going to be far more interested in what this head will do within the endothelial cell, and we'll continue this discussion in the next video.